Okay, so thank you for joining today. So today we have Dr. Amadou to present his research going very, very deep into genetic epidemiology. So today I think we have the opportunity to actually go broader on our questions. And well, earlier, just before, Ernesto was asking about which is actually the support of the distribution. I am translating it as the support, but I don't know if it's exactly the same. As the support of the distribution, then of finding a person with X value of cholesterol in their blood. And so how do you define the estimation, or how do you actually, how do you construct or build the distribution based on what you have observed, or you include what you haven't observed, which is actually the distribution that we are aiming for. Well, having said that, I just want to make a bit some comments that I think I just want to comment on popular things that have happened, especially something that for me is a bit dangerous, because, for example, here in Costa Rica, I got an email the other day offering me a loan, some credit, just to fly to the US and get vaccinated. I don't know about you, but I think it is a bit too much to get in debt just to buy something like this. But I think this is the space to actually question about these, and whether these social schemes are aiding or are helping, or what are they actually doing. And so it is actually, I think, well, for me, it is a very, very, very joyful day, because Amadou is one of my best friends while I was doing my doctorate degree, or getting my doctorate degree in Leicester Uni. And well, it was very good to have people like him around, and so that we could have endless talks and criticize on the edge of our doctorate and postdoctorate process. So thanks a lot for joining and meeting us on this challenge. And thank you, Mario, for being here. I know sometimes it's difficult, and Ernesto also, thank you for being here. And I know Amadou wanted to have like 100 people at the same time. But I think something that is going on is that our talks are being a bit too long. And so I think we have some of us that actually want to be on stage, and some of us, they are actually reading afterwards what we are uploading on the channel. So I think that's something that I think I feel very proud, because last time actually we had a lot of views on our last session. So that is actually something to be motivated and to carry on doing all of this work. So do you want to present your work to us then?
Yeah, let's go ahead. Did you finish with your announcements?
Yes, I did.
Oh, okay.
Thank you.
Maybe people would take a loan to get a vaccine. If the vaccine is fundamental to their work, they need to travel somewhere. If they don't have a vaccine and there is no vaccine where they are, I personally don't see a reason where someone would put himself on debt to get the vaccine right now, given how many vaccines are free. At least in the US and in many countries, you can have access to vaccine without much hassle. So I don't know what that is about. So I'm going to talk about polygenic risk score of kidney function in African American with hypertension. And first of all, I would like to thank Milena for inviting me to give this talk. It's challenging to give a talk to people who are not on your subject matter, but I view that challenge as an opportunity because when you are talking to people who are not in your field of research, that forces you to understand better what you're going to talk about and to use a language that is more accessible to the public. And I think for us scientists, sometimes there is a challenge of engaging with the public or giving a message to the public that they can understand in a language that is not too sophisticated. And my whole life, I think that has been one objective. Whatever I understand in my field, I should be able to talk about it in a language that is accessible to most people. And to me, that is extremely important when I sit in a talk with mathematical talk, statistics, and so on, and the person is just flipping equations and complicated stuff, and I don't enjoy it much. I want people to bring the subject to a level of language that most people can understand. Why are we doing this? Why is it important? What are the challenges? And kind of things like that. And keep the very, very hardcore technical stuff for the people who are deep into that field. So thanks, Elena. And I'm just going to start with the first slide here is just an outline. I'll talk about my background very briefly. Then I will go about some very basics of genetic epidemiology and genetics. And that also I included that because Milena told me it would be good to just comb through what is all this genetic epidemiology about. It's a very young field that branched out of epidemiology.
Let me just do a check. Are you showing the slides already, or is it on black board?
Oh, I don't know. Do you see the slides changing?
No, it's on white on blackboards. Maybe when you put it on a full screen, it goes a bit weird.
Sharing pause. Bring your sharing to the front. That's what it is saying here. Sharing is paused. Bring your shared page to the front. What does that mean?
Maybe you can stop sharing and share again.
Yeah.
Okay, then let's do that again. I'm going to stop it. Do you see it now?
Yes.
Okay, stop sharing. And now I'm going to share it again. Okay, now I'm just going to go there in the Zoom meeting and share it again. Do you see it?
Yes.
Okay, should I make it now full screen?
You want to?
Oh, I don't know. It's up to you guys. I just want you to see the slide properly.
Yeah,
if you see it,
we can see it.
It's okay.
Okay,
it's okay like that.
If it's enough big, then that's fine. Okay, so this is I will talk a little bit about my background. Then I will talk about some basics of genetic epidemiology, genetics. If any of you is a specialist in this area, I apologize. I use very loose language, very simple language that does not convey the whole complexity of this stuff. But I thought it's enough to stay at that level for maybe many of the audience are pure statisticians. Then I will brush about polygenic risk score, why we do polygenic risk score, and what it is. And then I will talk a couple of slides only on the analysis of doing a polygenic risk score of kidney function. And I will insist here on the motivation. Why am I trying to do a polygenic risk score of kidney function the way I'm doing it here? And then I will show you the results. The results are really, to me here at this point, anecdotal because this is still ongoing work. The results might change depending on what I'm doing and so on. but so far, what you can see as the results. So starting with my background, I grew up in Senegal. That's the western country in West Africa, below Mauritania. If you're looking into the map, the countries on the west, you have Senegal, and above Senegal, you have Mauritania. That is a massive land. And then above Mauritania, you have Morocco. Anyone doesn't know Senegal and Mauritania, maybe heard about Morocco. For some reasons, it's a very touristic area. I was actually born on the north bank of the river Senegal. That's a river that flows in West Africa, and it's in the border between Mauritania and Senegal. I was born on the north bank. Those days, there was just after independence, it's the same people living on the both sides of the border. These borders are artificial to us African. They were just borders that were made by the colonizer, who was France. So Senegal was colonized by France, and I was speaking French at a very young age, because the place where I grew up, there was a lot of French how you call it again, people that France sent for, what's the name again in English?
Colonizers.
No, it's some kind of cooperation in education where people come to Senegal and then they teach there and then they go back to France and so on. Where Senegal has deficit, they used to seek help from France, and France would send some expertise in that. So there was a lot of people from
Like
France.
exchanges.
Yeah, kind of, but teachers and professors and so on. So there were children who spoke French, and I played with them, and I spoke French before I went to school. This is very important. People who are autochthone, they don't speak French. They speak their local language. My mother tongue is called Fulani. Some other people's mother tongue is Wolof. There are about 15 languages in Senegal, but I do speak some of them. And I was fortunate maybe to speak French before I get to school, because if you don't speak French and you get to school, the first thing people do is learn French. So you have an advantage if you get to school speaking already French, because although it's not our language, the teaching is in French. So I grew up in Senegal. I'm showing you here a picture, a very nice city in Dakar, which is the capital city. You can see traditional fisher boats, and on the background is the downtown with high rise and so on. And I'm familiar with most of these places. I used to go there when I was young, and then you help them pull the boat, and then they give you some fish, and then you grill the fish on the beach there and so on. So that's me at the age of 11, I think, 11, 10-ish, just between elementary school and secondary school. And then I was very good at elementary school. And yeah, given my background, my parents never went to school. They're illiterate. My father left his village and came to city to just hustle. And I grew up in the city, and I went to school early, 2 years younger than the normal age. And then I progressed through elementary school fairly easily. And then I went to secondary school. Then I was just average, because also I wasn't studying hard enough. And then I went to high school. And in high school, this is a kind of the French system. When you get to high school, you choose your subject. And I was working towards a baccalaureate of physics, chemistry, biology, and math. Although my favorite subject when I was young was history, but I never studied history later. So in this high school here called Galandou Diouf, which is about 30 to 40 minutes walk from where I lived, I obtained a baccalaureate in physics, biology, math, and chemistry. And then I went to the University Cheikh Anta Diop of Dakar. It's one of, I can say, the best universities in West Africa, especially in some subject areas like science and medicine, particularly medicine. So I studied the year when I was 18, and I studied life sciences, or biology, physics, chemistry, and those kind of things, although I wanted to study medicine, but I wasn't good enough in math. This is really shocking. I went to Europe and I see people who don't even learn math, and they go and study medicine. but I wasn't enough good in math. And one of the reasons was when I was young, math was too abstract for me. I didn't like the way they taught it because I cannot bring it to reality. I just see X and Y's and definition domains, and I couldn't say, what is this in real life related to? So it was too abstract for me. It wasn't until I went to Germany to study that I realized, oh, if I can bring the math to understand it in the real life, then suddenly I start thriving in math. But early on, I wasn't really excited about math at all. So that's the university, that's the library of the university. It's named after a prominent African scientist called Sheikh Anta Diop, who worked a lot in proving that the ancient Egyptians were Black. And that's the whole his life. That's what he spends it on. He's well known in that field called Egyptology. So that's me here in the lab looking at cells of onion under this microscope. Then I dropped out. Actually, I never finished university in Senegal. The first year, there was a lot of trouble, political troubles, and students are always at the avant-garde of political struggle those days in Senegal. So whenever there was demonstrations or whatever political unrest, it came to the campus and the students used to struggle and so on. So they closed the university for 1 year. So here I am, come to university and lost a whole year. And then the second year I lost completely taste of learning anything. I'm like, oh, we are just come here. This university is not working. I'm getting nowhere. I didn't study properly. Then I dropped out of university. I just stopped going to university. And my dad was really affected emotionally by that because to him, he didn't go to school and I have the chance to go to university. And now here I am dropping out. Okay, then I started working in some non-governmental organizations. And then at some point, someone, really one of my mentors, convinced me, you need to go back to university and study something. So I applied to go into study in Germany. And a very good friend of mine, he's much older than me, but he was a very good friend. And he was wealthy. He decided to sponsor me to go to study in Germany. So I prepared the paperwork and I got an admission at the Mannheim University of Applied Sciences in Germany. So I went to Germany to study biotechnology. So in Germany already I was exposed to really gene editing, not gene editing those days, I would call it again, all kind of genetic manipulations and so on, and biochemistry and all of those kind of things. But then I really did not enjoy studying biology in itself and working on a bench, what we call wet bench, doing experiments in the lab, because I'm a bit of a dreamer. And when I'm doing Petri dishes, I might sterilize it and then put it somewhere, and then my mind is wandering. And then at some point I touch it again, and then now it's not anymore sterile. I have to sterilize it again, or I start putting drops, and in the middle of the drops, I think about something and I put 2 more. Did I count how many drops? Was it 3? Was it 4? I don't remember anymore. Then I have to redo the whole experiment. So I decided it's better for me to go into computer, because in computer, if you do a mistake, you just do undo and you go back to step before. So I worked in a company that does software business called SAP. I really become good at programming a little bit at that company. So when I finished my studies, I went to England. I worked a little bit computing and so on. Then I decided to do bioinformatics. Now, bioinformatics, as the name says it, is using informatics or computers to solve problems in the biological world, mainly molecular biology. So I did a master's in bioinformatics at the University of Leicester. And when I finished my master's, I was really, I think, the second best student on that master's. Then they offered me a scholarship to do a PhD in genetic epidemiology, which is a very good continuation, because when you study bioinformatics, you now have the computational biology skills. When you move into genetic epidemiology, then what you're going to learn is statistics and epidemiology, but you know already the computational side. So I studied genetic epidemiology under the supervision of Professor Paul Burton, which Milena knows, and that's how I got to know Milena, because she was doing her PhD there at the University of Leicester at the Department of Health Sciences. a very generous man, and I was even talking to him this weekend. And then he really supervised me, and then he introduced me to a whole new world called data science, where it's basically all kind of data, but you crunch them using computers, mainly biological data, but I developed really high level expertise in processing data. So when Professor Paul Burton moved to University of Bristol, he asked me if I want to come with him to do a postdoc at the University of Bristol. I agreed. It's University of Bristol, very nice city in the south of England. So I went to Bristol. I spent 1 year in Bristol, and then I got an offer from, and then I thought I need to change. I have been with my mentor for some time. I need some new challenges. And then I got a job at the National Institute of Health of the United States, which is near Washington, D.C., here in this little city called Bethesda. So that's my journey from Senegal to now here in the Washington, D.C. area in Bethesda. And that consumed a lot of the talk. Sorry for that. But I like to talk about where I came from. And if there are younger people in the audience to know that you might start late, but that doesn't matter. If you put your heart into it and you believe in yourself and you have some goals and you work really, really hard, you might get there, even if you start with a lot of handicap. So what
Just,
about
just a
genetic
quick
epidemiology?
question. Would you
Yes,
would
please.
you encourage other people from Africa or Latin America to follow your steps? Or which will be like shortcuts that you will take, knowing
If
what you
everyone, yeah, if everyone was to follow my footsteps, it won't be good for those countries because I call it a brain drain. People leave, they need the expertise in their country, and if they leave, then there will be not much expertise in those countries. I took these steps because at some point in my life when I was younger, I saw no opportunity back home. That's why I left. But if there are opportunities in your home and you are with your family, you are in your culture, and you can do it there, I see no reason for going abroad. You go abroad maybe to acquire different expertise and to have more experience and then to come back. But following my steps at a young age, leaving your country and then never looking back, kind of, or going just to work somewhere else, that might not be the best model for the developing countries. But at the same time, everyone has his own challenges and seeks opportunities the way they can. There are people in your country maybe have no opportunity. If someone gives them an opportunity to study something they want to study in Germany, well, of course, they will have to seize it. But I wouldn't say this is something that you would recommend to people. It came to me. It's not somehow what I said, oh, from the get-go, this is what I'm going to be doing. It's just lack of opportunity, please, an opportunity coming somewhere else. That's how I ended up here. And I want to say, if anyone has questions and they don't feel comfortable speaking in English, they can say it in Spanish and Milena will translate. So we talk about genetic epidemiology. This is what I did as a PhD. And genetic epidemiology is not really, it's using all the tools, the methods, the approaches of classical epidemiology. The only add-on is the genetics part. So Morton defined it as a science which deals with the etiology, distribution, and control of disease in groups of relatives and with inherited causes of diseases in population. You notice here disease in groups of relatives. This is an old definition because it talks about here how disease spread in families. That's why you see in familial studies, they build always the family tree and then so on, trio studies and so on. But really genetic epidemiology now is more of study of genomic variants that affect health and mainly now focusing, as you will see after, on complex disease. What complex disease are is just multifactorial disease. But this was one definition that I see encompasses and shows you actually this is really epidemiology. It's just that it has a genetic component of it because epidemiology deals with etiology, distribution, and control of disease in populations. So traditional epidemiology, of course, then explores environmental. We're talking about here in a wide sense, environmental. Diet, sleep, your lifestyle, all of those things I'm putting under environmental here. How those environmental factors and social factors, they influence the occurrence and the development of disease. That's what traditional epidemiology has been always doing. Now, in genetic epidemiology, we address the genetic cause of disease. And more now, this is really what most people do, try to understand the interactions between the genetic causes and the environmental determinant of disease. So, of course, then, because when we talk about genetics, it's all about DNA, genome, and genes. I put here a couple of things just to explain what those terms are. The deoxyribonucleic acid, DNA, is a nucleic acid that sits in the nucleus of the cells, as you can see here in this picture. Now, that DNA exists usually it's packed in chromosomes. The DNA, if you were to think what the DNA is, it's like a book that is coded into A, G, C, and T, which is adenine, guanine, cytosine, and thymine. These are the 4 basic fundamental bases that makes DNA. Depending on how they are rearranged, then what comes downstream would depend on the arrangement of the DNA sequence. So the DNA is packed into chromosomes as units. As you can see here, how a chromosome looks like. And there are 23 pairs of chromosomes for human. Depending on the species, the number of chromosomes would be different, could be different. So we have 23 pairs of chromosomes. Now, your whole 23 pairs of chromosomes, the whole DNA sequence of an individual is referred to as the genome. When we say 1 genome, 2 genomes, we're talking about actually the full sequence, DNA sequence of that individual and the full DNA sequence of another individual. And when you have, say, I have 1,000 genomes, it means actually the genomes of 1,000 people, like that study called 1,000 Genome Project. So in the genome, there are sequences in the DNA that are called genes. What are genes? Genes are segments that are coding, meaning they be translated into a downstream product, mainly proteins. So the genes in the genome or the genes in the DNA are sections that are specialized to produce something downstream. So because we're going to talk about gene expression, that's where we started in my analysis. It's important to know then what is gene expression. Gene expression is the process by which the information that is encoded in those 4 letters in those sections is then used to direct the assembly of usually a protein molecule. So you go from the DNA, then you go a transcription, you have a messenger RNA, and then the messenger RNA is, like the name says, it's a messenger. It tells the cell what to produce. Okay, so right now we talk a lot about mRNA vaccines. mRNA vaccines, messenger RNA vaccines. And I think what they're doing is basically inoculate or send a messenger RNA that has a specific message, meaning if I was to speak like, because what we talk about when a foreign body enters the body and the body fights back, it's like a war. So your body then would prepare some weapons to fight against the invaders. So here what they're doing is basically send a message directly to the cell. When it gets into the cell, the messenger RNA, then that message is transcribed and it tells the cell, hey, you need to produce X, Y, and Z weapons for an invader or potential invader that looks like some descriptions. So basically, the cell then would produce the antibodies necessary to fight this thing. Now, why is this revolutionary? This is revolutionary because for all of you guys, in epidemiology, before vaccine was kind of a weakened element of the thing that gives you the disease. You then inoculate it, and then the body say, haha, we know this attacker, and then they fight because it's weakened. Usually it never causes you the disease, but then the body recognizes it and then, come up with ways of fighting that agent later on if that agent comes again. Now, in this case, and the reason also I think they're moving into messenger RNA vaccines is because the conventional ways of making vaccines had a lot of side effects because they have to cultivate those agents in, I don't know, eggs or whatever. But they think if you tell the cell to make what it would have made if facing the agents, that might be more efficient and it will cut down on a lot of side effects and so on. And I think unless we have some unknown right now catastrophe that did not surface in the future, most vaccines would turn slowly into messenger RNA vaccines. So that's what I could say there. Don't quote me on that. If someone comes fighting at me, I'm not a specialist in this area, but because we're talking about messenger RNA vaccine, I thought you would just know from this in gene expression what it is doing. It's basically telling the cell to produce certain molecules. So this is gene expression. Now, anything happens, you can then imagine from this that anything that happens at the DNA level might then affect the messenger RNA, which might then affect also the protein. So mutations, I'll talk about genomic variants in the next slides. So mutations, in the colloquial language, mutations, what we call it, genomic variants, they're sitting on the DNA. They change the sequence of the DNA. So that might have an effect on the messenger RNA, which then might have effect on the downstream thing that is produced, which is here, we're showing proteins. So then what are genomic variants that might affect the end product of gene expression and hence affect health and disease? So people have slight differences between the genomes of individuals and the sequence. There are points in the sequence where there could be differences between individuals. And here is illustrated by in the Genome's website where I got this picture from. You see two people, and one of them has a C, which is a cytosine base in his DNA at that position. We call that position actually the jargon is loci at that locus. Then one person has a C and the other person has a T. Now, this is a difference. Now, it could be that the initial or the original, what we call the normal sequence, or the initial or just the original sequence has a C, but then there is a mutation that happened throughout life that caused some people to have a T there. Now, that T might be innocuous, it might not do anything, but actually it could also be a change in the DNA that that has some influence on health and disease. So this is a genomic variant. So from now on, whenever I say a variant, I'm talking about a genomic variant. So there are roughly about 4 to 5 million genomic variants in an individual genome. That might seem to be a lot for you, but then the total number of bases in the human genome, I don't know how many, is it maybe 500 million or a billion? I don't know. Don't quote me on that. But there are about 4 and 5 million genomic variants. But because the genome is extremely vast, so these 4 or 5 millions doesn't make a big difference between individuals, but they still exist. And these genomic variants arise throughout life and our interaction with the environment. Now, this is extremely important for you to note that different populations, I'm talking about here populations in the more of a geographic sense, people who evolve in Africa and stay there long and stay in Africa because of the interaction in the environment and so on, it shapes their genome. They will have certain mutations that are more particular to those areas than to other people. So then you will find then some genomic variants to be a kind of a signature of a population, okay? Now, if those genomic variants happen to be related to a certain disease, then that disease would be more prevalent in that population. Does that make sense? So what I said, and we're coming now into the disease and health. some variants increase the risk of developing disease, while others may reduce the risk, and others has no effect. So if you have a variant that reduces the risk of disease, we call it's a protective variant, meaning if you have that variant, you're lucky, you are more at risk of that disease. It's a protective variant. Usually that's what they use the language to say, just a protective variant. Here you have 8 people, and wherever you see a V is a variant, then you see people have different variants in different places in their DNA sequence. So then we go into diseases. There are some diseases that people have been studying for long, well known, and studied mainly in families that are called monogenic diseases. Monogenic diseases are diseases that are related where the disease is related to defect or variants in one single gene, hence the name mono. So there are a lot of monogenic diseases that geneticists do study. One of them that I put here, I think maybe one of the most common is Familial Hypercholesterolemia Fh, and it is caused by having some genomic variants that puts you at risk in a gene called LDLR or a gene called alipoprotein B or PCSK9. So these are well known. If you have those genomic variants, in those genes, it disrupts those genes. And what follows is known as you have a very high cholesterol. And if you have very high cholesterol, it can lead to blockades in your coronary arteries and so on. And here I put some signs of how people would kind of suspect that someone has FH, and because they will have a very high LDL and all of these signs. But the ultimate test is a genetic test. They can just test and see if you have those genomic variants. If you have those genomic variants in these genes, then they will know, oh yeah, you have high LDL, you have a family history, you have all of these signs. Plus, now we have confirmed that you have this genomic variants or in any of these genes. So that's the disease FH. Another monogenic disease is cystic fibrosis, which is well known also, where some fluid, I think, build up in your lungs and so on. So these are less challenging to study because you know at one position, if the person has a genomic variant, then if the condition is dominant and so on, I don't want to go into those details, but most likely if you have those variants in that gene, then you have a higher risk of having the disease, or you will have the disease depending on the penetrance. So what we really more focus, or at least myself in my career, I'm focused on is complex disease. Now, complex disease, as the name says, they're more complex, but I prefer the name multifactorial disease. Multifactorial because there are a lot of factors that came into effect. One of them is the genetics, and that's what we are studying. Part of it can be environmental. It can be also lifestyle. Do you smoke? Do you drink alcohol? The environment could be also your social environment and everything. So all of these factors together combine then to increase your risk of a certain disease, okay? Let's say, I'm gonna give you an example, PTSD, okay, that people come for when they have a trauma. Let's say someone has some genomic variants that increases their risk of PTSD. Now, that does not mean that they will have some PTSD, but if they go through a trauma, maybe they went to war, maybe they witnessed some massacre, those people are more likely to develop then maybe PTSD than the person who does not have that increased risk related to the genetics only. And you can give, that's where the environment or something what happens, okay? And another one is sickle cell anemia. People might have the trait or might have the genomic variants that puts you at, that gives you sickle cell, but until you go into high altitude where oxygen is rare or there is some trigger, you might not experience a crisis, or the disease might not manifest into you. So sometimes the environment can trigger something that was existing in your DNA, but would have not maybe caused you any harm, unless you are in a different environment that then increases your risk or act as a trigger. Now, complex diseases, people study them using genome-wide association studies. You hear the word GWAS, that's what GWAS is. What they do then is usually, if it's a binary, like you have the case control, they will just take two groups and then of people who have the disease and people who do not have the disease that are controls, and they see the prevalence of the genomic variants. If the prevalence is significantly higher in the people with the disease, then they will say, oh yeah, these genomic variants are related to this or are correlated with this disease. If the genomic variants are more prevalent in the controls, they will say then, oh, there are some genomic variants related to this disease, but they seem to be protective. Okay, because people who have the genomic variants tend to not have the disease. Okay, so that's GWAS. Now, people have been doing GWAS over the past decade. They produce a lot of data. So every time you see this mutation is associated with this mutation associated with disease, but we don't know the mechanism. We just know this is a correlation. Statistically speaking, it's a correlation. Until we can tell what is the process. Oh, actually, this genomic variant is sitting on that gene in the promoter of this gene, or it is sitting in the regulator of this gene. The gene cannot be regulated, then it's downregulated, or it's too much upregulated, and you're producing too much of this downstream product, and that causes you this disease. Until we understand the disease, the mechanism is mainly a correlation, but a strong correlation, because to rule out false positives, they put the p-value has to be 10 to the power of -8 for them to believe that this is not a false positive, because it's a GWAS. You're combing through millions of genomic variants, then false positive is bound to happen. So you put a threshold to very high to filter out false positives. So in these GWASs, then I'm talking about now polygenic risk score. What is a polygenic risk score? A polygenic risk score is because certain diseases have many genomic variants that are related to them. So when we want to know if we combine those variants, because most of the time, each of those variants doesn't have a very strong effect, but what is the combined effect of all of those variants? So you put those information in a computer and you use statistics to then know what is the relative risk for that individual. Kind of you have a score for the individual, like that, in this chart here from the Genome Project. You can see that if you calculate a polygenic risk score, some people would have a high risk, some people would be at low risk, and most of the people would be at the middle. What we're interested in is who is at high risk and who is at high risk and who is at low risk, depending on the combination of the genomic variants they have. So that then it's not an absolute score. It's not an absolute risk. It's really a relative risk. That's important to make a distinction here. So how do we do a polygenic risk score? First of all, you have to find genomic variants that are associated with the disease. How you do that? You can go into GWAS. People have done a lot of studies of let's say, hypertension. Then you say, oh, these people have found this genomic variant to be associated with hypertension. You take all those genomic variants that are said to be associated with hypertension. Then you say, I have selected genomic variants associated with hypertension. You want to now combine them. Then the next step is you obtain the weights, because a polygenic risk score is a weighted sum of the alleles. Each individual would have two alleles because the DNA is, you have two sets of DNA, one from your mother and from your father, from your dad. Then how many alleles of the genomic variants do you have? That then is multiplied by the weight. The weight is nothing but the effect. We're going to talk about kidney function measured as eGFR, glomerular filtration rate, estimated glomerular filtration rate. So if we were talking about systolic blood pressure, then it would be the effect of the genomic variants on systolic blood pressure. Does it increase systolic blood pressure? Does it decrease systolic blood pressure? That effect size is the weight of that genomic variants. So now you have the weights. Usually you go into a larger study to make sure that the weights are robust. So, and then you build your score, which is a sum of the risk alleles weighted. Then you have your score for each individual. Then you test the score, which is basically just doing a regression or doing a t-test, looking at the people at the tails of the score. Are they significantly different? So that's how you build a polygenic score in a very simple layman terms, although there are a lot of challenges about the weights here. We don't talk about that. Now, why am I, or why are we interested in polygenic risk score of kidney function in African Americans? First of all, we have to acknowledge the fact that most of the genetic data that exists is right now European. I'm showing you here a chart from the genome. There are different versions of this chart all over that has been published. Most of the genetic data to date is from European populations. And you can see 10% is from Asian. Now, you might be fooled. This Asian here, most of it is from China, because recently China has advanced in sequencing, and then they sequence a lot of populations and so on. So this 10% is not very diverse. It's mainly from mainland China, okay? Not the rest of Asia. Then you have about 2% of African, 1% of Hispanic, and some minorities, less than 0.5%. And then for 8%, they don't know what their ancestral background is. Now, this poses a challenge. If you want to do genetic studies and most of the data is on some populations and not on other populations, that's a big challenge. So you can't do, it's difficult to do a polygenic risk score in some populations because nobody has studied those populations and they don't have data in the genetics. And that's one of the issues I'm faced with doing kidney function in African American or in African, because most of the US, those big studies that tell you that this genomic variants are associated with this disease, most of them were done in pretty much Europeans. So this is a lack of diversity in genomic studies. It's not very diverse. Because it's not diverse, it can, at some point, if it's not addressed, cause health disparities. If we're developing solutions, gene therapy, or we go into precision medicine, and most of the data is from European data, then who's going to benefit from those solutions, health solutions, then it will cause, again, health disparity between people who have European background and other populations. So another motivation for doing this analysis, and this stems from what I said before, because most of GWASs are in Europeans, then you have a problem, because when you calculate a genetic score for a European population, you cannot transfer it to an African or maybe even to an Hispanic population. Why? Because genetics is population specific. Remember what I told you before, your genome is made through your interactions with your environment. So then there are some patterns that are known in certain populations more than in other populations. So if you have some genomic variants that are more prevalent in a certain population, you calculate a risk score there. The prevalence of those genomic variants is different in another population, so you cannot transfer the score. And this paper really talks about that in terms of kidney function, in terms of kidney disease, and saying that basically kidney disease polygenic risk score is not transferable, first because the disease is very heterogeneous, secondly because most of the data is from Europeans. And then in that last point that I'm putting here, they said they think the solution is for people to do these studies in those understudied populations. So African American or African or most of the minorities are understudied populations. We decided then to do a polygenic risk score in African American because it's a population that suffers a lot from kidney disease. A lot of people die of kidney failure and so on, cardiovascular health. So then finally, the last motivation, and I already kind of alluded to this. This is a very nice paper by colleague Alicia Martin. And she showed that actually, if you have a polygenic risk score in European in that plot, and you try to predict the outcome in other populations, you can see how the prediction drops from European to American to South Asian to East Asian to African. So basically, you have a score that should tell you where the person sits in terms of risk, but then it tells you the risk very well in European, but you can't use it to predict the risk in African. So again, this is another reason why we're trying to build a polygenic risk score that is specific to African populations, and you can do it in Latin populations and so on, because it will help to know what genomic variants increases the risk of disease in that population for people of that population. or people of that genetic makeup. So this is the strategy that I started. I'm going through it quickly, but you will understand it because we have already now talked about the basics. We talked about gene expression. So I did not go into GWAS to do this first step here. This is the first step, find your set of genomic variants. Now I have to select some genomic variants to start with. Now, there are no big study there of people of African descent for me to choose select variants from. So I decided to select from the data that I have because I have the gene expression of 78 African Americans, meaning I have their messenger RNA level for across, I think, 50,000, 25,000 genes and so on. So what I decided to do then is I use machine learning here, random forest, to say, can I find genes that can predict EGFR, which is a measure of kidney function in these 78 people? See, I'm working back. I find the genes first, then I find genomic variants that affect those genes. And those genomic variants that affect the genes that predict EGFR, they are then my selection of genomic variants. So first, then I find genes. Actually, it's messenger RNAs. I find all the messenger RNAs. I'm just using the term genes because usually we use them as synonymous of messenger RNA, but it's not the same. So I find all the genes that can predict kidney function in these people. Now, this sample might seem to be small compared to the other sample, but this is gene expression. We're talking about here 50,000 data predictors or 25,000 predictors. In my case, 14,000 genes. What I'm telling the machine when I run random forest is, can you find me a subset of genes that can classify with a very good area under the curve who is at the high end of EGFR and the lower end of EGFR? So then I found 3,493 genes that can predict the top tertile and bottom tertile with an area under the curve of 0.86. Okay, so then I now have genes. The next step then was to say, can I find then genetic variants? And they're called EQTL. This is expression quantitative trait loci. It's just a name to say a genomic variant that affect the expression of a gene. So I found 22,000 genomic variants that affect the expression of 2,500 of these 3,000 genes. Now, those genomic variants, they're the one that I'm going to use into a polygenic risk score. Before that, I've jumped here a step. There are steps that I'm going to refine this list here, tighten the list to the most robust predictors of the expression of these genes. Now, that subset of the most robust predictors of the expression of these genes that predict EGFR, they're the one that I'm going to use in my polygenic risk score. Then I take the weights from UK Biobank, which is a study in the UK. I take all the people who have African ancestry in the UK Biobank, then I say, what is the effect of each of these genomic variants on kidney function? That's my weight. That's the effect size. It's just literally a regression where the point estimate, I take the point estimate as the weights. So then I combine the point estimate with the number of alleles for each individual. Then I have a score. I test the score in the ImageGrid, then to see if the score is associated with kidney function. And then I validate it in an independent data set called regards. So this is the data. I'm not going to dwell on it too much. The ImageGrid, this is the distribution of EGFR, age, they're younger than the UK Biobank, sex, this is male, female, this is systolic blood pressure, diastolic blood pressure, and BMI of all the three studies that I'm using. The ImageGrid is the Minority Health Genomic and Translational Research Biorepository database. It's African American from the southern part of the US. And regards also, it's a study of stroke. And I also abstracted thousands of people of African Americans from that study. And in the UK Biobank is a study of 500,000 individuals in the UK. Among them, there was about 9,000 people who declared to have some African ancestry. So I picked those people too. And then before you build, when you all know that if you test something, you have to validate it in a population that is similar. Now we're talking about here genetics. Look here on the top left. Each point is an individual. And these individuals here, they have declared their ancestry. Some of them say they are mixed, black and black British. Some of them say they're white and black Caribbean. Some of them say they're Caribbean, like Jamaican, and plenty of them in the US, in the UK. And some of them are African people who immigrated from West Africa countries like Nigeria and so on, colonized by the UK, by England. And the black dots is my data. It's image grid. So if I project my individual from image grid into UK Biobank, you have this dark cloud here. So that means there are a lot of people in the UK Biobank who say they have African ancestry, but they are far away from these African Americans in terms of genetic makeup. Okay, so I'm interested only in the people who are similar to the African Americans in terms of genetic makeup. So then what I'm doing is do a matching. I match them on this is a principal component analysis plot. I match the people by the first principal component, the second principal component, and the third one. So then you see the red dots here. These are all the people from UK Biobank that were matched to the people of image grid, my data. And I did the same regards. Regards was pretty much not different from image grid because they're all African American, but you can see here, I don't want to go into the details. On the x-axis, it's like you're moving from Africa to European populations. So the more you are on the left here, the more you have African ancestry. The more you're on the right, the more you have European ancestry. So in regards, there are people who maybe have a higher European ancestry than African ancestry. Then I weed out those. So when you go into the plot on the right, now I match again regards to image grid. So I have people who are similar to the image grid to do what I want to do. Okay, these are the results, nearly the last slides. Then I built the score. And then I look into the association between that score and EGFR in a linear regression. And I looked also in a t-test where I compared the people at the bottom of the score, the lower quartile to the top quartile. I compared those two groups. So here, what I'm seeing is that the number of genomic variants in the score is 137. I built a score with 137 genomic variants. What I see that is that score is associated with a decrease in kidney function in image grid, and I validated that in regards, it's also associated with a decrease in kidney function in regards, meaning those people who have these genomic variants, their kidney function would decrease. The higher your score, the lower your kidney function. So, and that you can see also in the t-test here, when I looked into the people who are at the top quartile and the bottom quartile, I compare them, you can see that the people at the lower quartile have a larger score and a larger EGFR, which is a measure of kidney function. 99 here for people at the lower quartile and 90 for the people top quartile. And you can see also the same trend in the regards. So to finish, basically what we have done, this is the acknowledgement to Milena, to the Image Grid Consortium, to UK Biobank and regards. Basically what we did here was there is not enough data out there to do a polygenic risk score of African Americans. So we use our own data to find genomic variants that affect genes. And then those genomic variants are our selection that we use to build a score. We find this score to be negatively associated with kidney function, meaning the higher the score, the lower the kidney function. And we tested it in our data and we validated in an independent data set. So that's what I wanted to tell you about today. Sorry, it's probably a lot, but I try to keep it as simple as possible without violating the language and everything, but I'm sure I did a lot of cookups and approximations that specialists might not be very happy with.
I'm really thankful, Amadou, with you, and I think it was a very, very great talk, and it has been very clear. I think it's going to be very useful in order to actually learn basic things of genetic epidemiology. I want to raise some questions. Some of them are like actually challenging into the value of the information. And one of my questions is, I mean, in terms of publication bias, how would you estimate? Because this is what I am kind of spotting in all of these. I mean, there isn't enough information for certain risk scores. And having said that, which will be the challenge for decision making in the use of this information, or will be the benefit of carry on and collecting more of this data. It is interesting to me, actually, the case of Asian, and that you actually said that this is recently raised in data collection from China specific. Is that what I understood well? Or what is it going on with this information? Because I think with depending on the country that you're looking for, or that you are this data actually is not available. For example, in Costa Rica, the problems that we are experiencing at the moment is that all of these challenges with the budgets and things have stopped some of the data collection of the university, and we have been lost. I mean, the window to collect some data that could be very valuable. But I think there is a meaningful question behind all of these, and in terms of where are we heading in terms of knowledge construction, or building up knowledge in terms of what we are learning from this field of genetic epidemiology.
Okay. First of all, in terms of bias, the bias do exist, because even in genetic epidemiology, people tend to publish only what they call positive associations. If they find something not associated, they usually don't get it published. So that means when you're going to find variants, you find basically things that corroborate something. But then if someone did an association between certain variants and, let's say, hypertension, and he find no association, but the study is robust and everything, usually they don't get it published. And that's important information, because we know then, oh, this area of the genome is not associated with hypertension. There's no need of looking into there. But if that data is never reported, it's not published, because you have to have a significant association and so on, there's a publication bias, of course, because people publish only things that are associated. That's one. And in terms of data collections, for genetic epidemiology and genetics, it's slightly changing. Why? Not because people suddenly has become charitable and they decided, oh yeah, these poor populations, we have no data on them, let's put money in and collect the data there. No, because they realized we are all the same human species. If you need information in genetics, you have to go back to where people lived first on Earth, which is Africa. And some variants that exist only in Africa, because some populations never left Africa to migrate to populate the rest of the world, you have to go to where those variants exist. So there is now some effort to go into Africa and some populations we have never had genetic data on, and to sequence them, because those people has been, maybe, let's say, just example, they has been in touch with some infectious disease that has not happened maybe somewhere else in the world. Now, their genome has evolved by being in contact with those infectious disease, or where the infectious disease is prevalent. That tells you something about what you observe in the nowadays. And the proper example is, for example, for kidney disease, they find a variance, I think about 7 years or 10 years back, a couple of variants And when they went into investigate them, they realized that that variant is a protection for African sleeping sickness. So population developed that variant to survive sleeping sickness in Africa. So now you have the historical context. Now, people developed that, it was protective. Now, it has some side effects, like just every medicine. It protects them from sickle cell disease, but then it puts them at more risk of having kidney damage. So then they see a lot of African American or people from that continent having a lot of kidney issues. Some of them, that kidney issue is from genetic variants that thousands and thousands of years back evolved or rise because as a way of adapting to an area that is where sleeping sickness is prevalent, where that fly, let's say fly, is very prevalent. So I think as we move on, people would understand that it's beneficial for all mankind to collect genetic data in all populations. Now, when would they understand that? Of course, the scientists are only interested in many, maybe publishing their papers and so on, but those who have that humanistic view will understand this field would benefit better if we start collecting data on all the populations so we know all the genomic variants that exist in human beings and not genomic variants that exist only in a certain population. Now, this might be the case in Latin America. It could be that they collect on more of the Latino who have some European background, and the indigenous people, they will be maybe sequenced last, because as before, they used to be, still it's very expensive to sequence the whole genome of one individual. So those who make the decisions might decide to sequence certain people and not other people. And that would cause also a disparity. And you guys know maybe Latin America or the whole of Southern America better than me. I can sense that if they are doing a sequencing of populations there, the indigenous people who are far away, maybe from urban areas and so on, would be the last to be sequenced. So collection of data then would be again biased towards certain populations or certain groups.
What do you think, Ernesto?
I have a lot of questions, but I have only 5 minutes because I have to leave. But let me, unfortunately, I have to leave, but let me just to say something now. The first thing to say is that this is a good example in which we need to make a differentiation between, let's say, an inference or a data description, whatever, which is overall or which is conditionally on some specific, I don't know, characteristics. So what happened, for instance, what happened in Africa or whatever is conditionally on that. And sometimes we make some comparison between a conditional distribution given, I don't know, some specific characteristic and either a marginal distribution or other conditional distribution. So, and I mean, it will happen that we will realize difference because, I mean, because the distribution are different. I mean, so the other more, let's say, philosophical question is that at the end of the day, you are defining a disease as I don't know if I am if my words are correct, but a disease is a kind of deviation from some normality, and the normality ends, let's say, when some variants appear. But I don't know if it is a good way to characterize a disease or not. And in particular, because you say most of the let's say, data or information we have come from Europe, and maybe we are defining a disease with respect to a specific population, and not necessarily this is a disease. I don't know. So
Okay,
I am.
yeah, I hear you. Maybe I did not explain it very well for you to understand. The disease is not defined with respect to a population. The disease is disease. Let's say hypertension. There's a definition for hypertension. It's a consistent elevation of blood pressure above a certain threshold. If you are white, black, Latino, or whatever, if your blood pressure, systolic or diastolic, is above a certain value for a long time, you are considered hypertensive. Okay? So the definition is the definition. Now,
Yeah,
it
but
could
you
be that
provide me an example, but you are providing me an example which is an operational definition. And if somebody change the level of pressure, the disease change. So it's not an essential definition. It's just an operationalization.
Okay, okay. Okay, but there is a definition. In medicine, we have to define a disease, isn't it?
No. In medicine, you provide an operationalization. Not necessarily it's a disease.
Okay.
Not
What do you
necessarily.
what would you consider someone with a systolic blood pressure of 190 over 110? Would you consider that a normal blood pressure?
No, it's just an operationalization. It's similar.
No,
when
if you are a doctor, someone comes in and you measure them and their blood pressure is 200 over 110. As a doctor, you have to do something. Tell them they are at risk of stroke because their blood pressure is too high. Okay? When we are talking about defining disease, we're talking about a clinical definition. It's not me as a statistician. Like ATP3 would say, if someone has an LDL cholesterol of 200, he is at risk. Okay? Then we have to do something. Then we say, okay, what is the safe range of LDL? They will say, okay, from what is observed in clinics, the safe range of LDL is this. We're talking about here only disease that are defined by the measure of a continuous variable like systolic blood pressure, LDL. But there are other diseases that are whether you have a stroke or not. If someone had a stroke, we cannot say he didn't have a stroke. Okay? Let's just go into the binary disease now. Whether you had a stroke or you did not have a stroke, you take 1,000 people that had a stroke, okay? They went to clinic because they had a stroke and they scanned their brain and everything, and they have the symptoms that goes with stroke. They nearly maybe died. That's one group. Then you take a group, never had a stroke, same age, maybe same context and everything. Then you look into their DNA. You say, are there some mutations that are more prevalent in one of these two groups? After we adjust for all kind of things, the two groups are similar. We can say this variance is more prevalent in the people who had stroke. And then we can go back to families. We say, oh, actually in this family, this man's father had a stroke at 55. His granddad has stroke at 55. We look at his DNA, we look at his dad's DNA, we look at his granddad's DNA, all of them has the mutation that is linked to stroke.
So the two group, you say me the two groups are equivalent, but at the end of the day, they are not equivalent because some of them
we are trying
variants and others not. So
no, okay, that's what we're trying to find. That's what we are trying to find, Ernesto. We are trying to find what is the difference between these two groups in their DNA. Simple. If we were to simply put the question, we are trying to find what is the difference between these two groups. Is there a difference in their DNA? That's the simple question we're trying to answer. It's like saying, oh, those people who have COVID and people who went into ventilation and nearly died versus the people who are asymptomatic. What is the difference between those two groups? You can ask the question in terms of serology and so on. You can ask the question in genetics if you think there might be a reason why some people, they have COVID and they are symptomatic, while other people, they have to go into ventilation. They have the severe form of the disease. These are the questions that we are trying to answer in genetic epidemiology. And it is now answered for certain disease. We have seen clusters, some disease are more prevalent in certain populations because the mutations that are related with that disease are more prevalent in that population. So we are not defining the disease by population. The disease is the disease, but we want to know what is the genetic background or the genetic underpinning of that disease. That's what we're trying to understand.
It will be nice to discuss, but I have to leave. So if I don't know if Milena could organize a meeting for making discussion, because in order to understand the logic of the.
I think what is really interesting of this discussion that is emerging is that it's clarifying that there is another question behind all of these, which is actually how we define disease or even how we define health in terms of how we specify the models that we are observing.
That's an excellent question that goes beyond the genetic epidemiology. Who decides what level we put the threshold to say disease, no disease? That's a philosophical question we need to ask, has nothing to do with genetic epidemiology. In all fields, we need to ask ourselves, are we defining it right? Because 20 years back, they would say, oh, if you have a blood pressure above 140, you are hypertensive. Now they lower it to 130. So you see, so the decision being made there, that decision can be discussed. How we define disease, that's really something. In certain areas, of course, we need to define what we mean here by disorder or disease. Unless if it is binary, if you have a stroke, we cannot say you didn't have a stroke, you had a stroke.
Well, but it's how you define the stroke also, on which conditions that you actually have, or
well, if you scan someone's brain and they have something popped and the blood goes in then and their mouth is like this and their hands is like this, would you say that guy never had stroke?
But that's the event. I mean, which is because if we are talking about genetic epidemiology, is that of how I think about this is that we are trying to link the causality of this disease with genetics.
Yeah.
And it might not be, I mean, I think
if we don't,
I remember one time having a talk like this, but like 10 years ago. And it was like, we, at the beginning of genetic epidemiology, maybe, or something like that, that people or even countries were confident that genetics information were going to be like, okay, this disease is because like that. I mean, it's gonna be like that detail.
No, that's true. That's true. Yeah, that's true for monogenic disease. Like BRCA5 for women have breast cancer. Those kind of things has been established. If you have these mutations, you are at a very high risk. And usually they will observe in your family, your mom and so on had either breast cancer or ovarian cancer or so on. For some monogenic disease, that's the case. But for complex diseases, cancer, diabetes, is extremely hard. And that's why people are doing polygenic risk or this and that. This is in its infancy. What we're trying to find is basically, is there an indicator of higher risk in your DNA? Now, of course, as you said, how we define disease becomes extremely important, because if what we are defining as disease is not disease, that we mean we are misclassifying cases and controls already.
Well, this is when I say that maybe we should, like, for example, here in Latin America, that we are, I think this COVID situation has brought.
Did you stop recording at least?
No, I will still recording. I think we're still
recording. We're talking about, maybe we are talking crap here, so don't record it.
It's okay because this is part of the deal. I mean, a lot of people are having these questions, and I think we need to talk. That is the point, basically. We need to actually understand, and I think it is easier to understand, especially when, let's talk about infectious disease with COVID-19. This is probably one of the type of disease that that we as a world overcame like very long time ago, and now we are having this problem. But which are exactly?
They're calling me for dinner.
Okay, so I'll leave you in a sec.
Yeah, I mean, what is important for us scientists is to stick to the data. What is the data saying?
Yes, but I just
let me finish
what I was, just the fact that how are we defining, for example, COVID-19, and which is actually one thing is actually the definition, one thing is what happens at practice, one thing is what the medics and the people are actually acknowledging that this definition is, and plus all of the prejudices and things that might come across.
Yeah, I mean, we need to keep the message very simple. Have you been infected with the COVID? Have you been in contact with the virus? That question we should be able to answer, because if we check your blood and you have antibodies, you have been in contact with the virus. Now, have you developed a severe version of the disease, or were you asymptomatic, or blah, blah, blah? That would depend. Someone would have runny nose. Someone would have nothing, absolutely nothing, but he has been in contact with the virus, because we find weapons that his body developed to fight the virus. And the virus did not just happen there, and then his body started, some by magic, creating antibodies. If we check your blood and you have antibodies, then you have been in contact with the virus. Now, what do we do about that?
So
that's
there
another question.
There is the point that I want to give you just to close this conversation, which I thought it's been very, very interesting, and it's been very, very useful. And the key thing is that we cannot look at things as a simple thing. One conclusion will be that, like, if we look at only one gene, for example, or only one thing, we are losing perspective. We need to go ahead and step across all of these challenges that we need to take into account, like environment, and I think in this sense, to be patient and collect the data, just be careful of the quality of the data, and then just be patient to observe results to actually acquire some knowledge on this.
I mean, there are two times, you know this. If you are a healthcare, you are responsible of healthcare, your mindset is different from the scientists like you and me, who can sit there for months researching something, trying to nitty-gritty. I am different. Maybe a clinician, he wants to just know, how can I solve this issue? I'm having patients coming. His timing is different. So he cannot function like me, a researcher who can sit here and, like right now, I've been working in kidney function, polygenic score for more than a year. Now, if this was a crisis, they'll tell me, go to hell. We need a solution. What are you doing? We cannot just sit here. You're telling us all of this complex stuff. We want a solution. Okay? Like when the water spilled, I think the BP plant blew, and then the oil was flooding in near Venezuela or whatever. If some complex guys come and start saying blah, 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 they'll say, come on, how can we solve this, or at least mitigate the issue as quick as possible? The emergency decision is not always the best. We have to accept that. Because emergency decision is guided not by, it has to be the best. And it's guided by just let's stop this as quick as possible. And later we find the way we stopped it is not the best. But then we have to continue the work and see, what have we learned from this? So next time a crisis happens, we have learned something, and then it will improve the way we react. That's how I see it. Because when a clinician comes in the ER emergency, someone comes, the way he makes decision might not make sense for me because it's not the best informed decision or whatever.
Yes, well,
Anyway, I
this
have
is
to go
how
now.
why I will encourage slow science and also maybe also slow decision making. Thank you,
Okay.
Amadou.
Thanks. Bye.
Thank you for everything. See you later
You're-
then.